Hello class, welcome to the second part of the remote lecture, pre-recorded lecture, lecture 6. So we just wrapped up the phase response curve stuff, and we're going to be talking about the dual oscillation model for this circadian clock. All right, so let's get started. So as we've seen some before in previous lectures, there is some kind of peculiar phenomena that occur when you bring in this when you bring the circadian rhythm into the laboratory and really start messing around with things like the light-dark cycles and the period length of the cycle. Things like really long or really short cycles, trying to entrain animals to something like 27 hours or 22 hours, or really weird light regimes like light, or what they call skeleton photoperiods, where they just get just a little burst of light twice a day that kind of represent the beginning and the end of the light period, things like that. And so we kind of see some odd things just from people trying to understand what is the circadian rhythm and its different features. And so from all this weird observations, there was an effort to generate some kind of conceptual model that could explain all of the weird things that they were seeing. So what are some of these weird things that I keep mentioning? One of them is alpha compression. Now, if you remember, alpha is the length of the active period. And so they saw alpha compression, which is the reduction of this length of the active period during circadian cycles. And so, can you remember, where have we seen this before? Can you remember in a previous lecture, 2 or 3 lectures ago, we actually talked about, we described it as a rule that we see in which alpha levels are generally compressed. Right? Do you remember the circadian rule? So part of the circadian rule is that intense constant light causes an alpha reduction in nocturnal animals. Right? It does the opposite in diurnal animals. It causes an expansion of alpha, a lengthening. But in nocturnal animals, which are the ones that have been studied the most in circadian rhythms, we see an alpha reduction when they're exposed to light. And we also see alpha compression in the laboratory when we try to entrain animals to cycles that are different than 24 hours. On the left, you can see an animal that has entrained to a 24-hour light-dark cycle. This is a double-plotted, 48-hour plotted actogram. And then on the right, we have an animal that is exposed to a 26-hour cycle. Right, and it's slanted here because the window here is still a 48-hour. The width of this image here is 48 hours. So that's why it all looks delayed. But you can see, you can use your eyeballs, and you can see that there's a clear compression of the alpha phase. The black ticks are much narrower than in the 24-hour period. Right, so this is an interesting phenomenon. And we also see alpha compression in something called skeleton photoperiods. Right, so at the very top of this graph, it's sort of a normal or somewhat normal light-dark cycles. You can see from the bar at the top. It's a long day length, but there's a clear night section, and they soon start with with the skeleton photoperiod. So the light that they exposed to the animal only comes on at the very beginning of the light period and the very end of the light period. So actually the middle of it is dark, right? But it's just the beginning and the end. So that's why they call it skeleton. And as you can see over time, as we go down through the days, they actually widen the skeleton light period, so that here that our night period in the middle gets narrower and narrower as the end of one photoperiod gets closer to the beginning of the next light period. And so, as you might imagine, the activity gets narrower and narrower. And then at some point, they see that all of a sudden there's this jump in the circadian rhythm in the active period. And this rapid phase jump by the oscillator seems to function to put that rhythm in a more acceptable phase relation to the light pulse. It sort of is like seeking the dark period in a weird way. And so the question is, what kind of structural feature of the pacemaker could account for this ability to compress and then all of a sudden decompress and jump somewhere else in this expressed locomotor activity? In addition to phase compression, alpha compression, we also have after effects, which we've seen before. Animals coming out of light periods into dark. They often have free-running tails that are elongated or extra long over time. They come back to their normal free-running period length. And so these after effects are seen in other lighting regimes as well. And so entrainment to different lighting cycles often influence the period length of that free running during dark periods. And so this example on the right, we have an individual that has two different period lengths in their free running rhythm. And we can see at the bottom when they're exposed to 18 hours of light. So this is actually a triple plotted, a 72-hour period, or 72-hour plotted actogram. And so this big giant block, this big square, is 18 hours. So you can see there's two 18 hours here. And in between those two is the dark period. And they entrain to these 18 hours of light pretty well. And then after they're released into dark, they have a fairly short tau, free running. So it's really angled to the left pretty severely because it advances quite a bit every day. So it's less than 24 hours. It's shorter than 24 hours. And also, as we'll see, there's some alpha compression as well. Compare this to what it had previously, and it was exposed to a day that was just 1 hour. Now don't get confused here. This isn't a skeleton photo period. It's just you're seeing two different days here. And so this is a 1 hour of light during a 24-hour period. They entrain to it, and when they free run afterwards in dark, you can see that angle to the left is less severe. So their tau is running closer to 24 hours, following 1 hour of light per day versus 18 hours of light per day. So this light exposure has these after effects. It seems to be influencing the length of tau. Right, so this is called an after effect because it persists. And it suggests that the oscillator system is susceptible to modification by previous experience. These types of after effects follow exposure to different day lengths, may have important implications. Right? Because many organisms normally experience a range of day lengths during each annual cycle. Depending on where you live, if you live at extreme north or southern latitudes, your day lengths are going to differ quite substantially. And in fact, some places, if you're really close to the Arctic or Antarctic Circle, you would indeed experience things like just 1 hour of daylight a day or 18 hours of daylight a day, or in even more extreme, such as 24 hours of daylight a day for part of the year, where the sun never sets. So what feature of a pacemaker system could endow it with this kind of, quote, memory, so to speak? So one of the most dramatic phenomena that we've observed manipulating circadian rhythms in captivity is this rhythm splitting, which we've seen before in a previous lecture. And so this was first observed in a tree shrew, Tupaia glis. So when this tree shrew was kept in light conditions, and then that light was reduced, that previous unitary rhythm, that single rhythm, split into two separate components. And that temporarily expressed, they for a brief time expressed different period lengths. So the two different oscillations had two different taus, which you can see at the transition, right at the transition from when it's one rhythm at the top to where it splits into two. You can see that one that comes off to the right seems to have, it's a tau greater than 24 hours. It seems to have a delay. And then when that second rhythm first split, it actually looks like it's got a bit of an advance, so it would be less than 24 hours. But then they eventually parallel each other and run parallel to each other 12 hours apart, right? Or you could describe that as a 180-degree phase angle. And so this was, surprising, as you might imagine, to discover this at the time. And then once the animal was exposed to bright light again, those two oscillations fused back together. So despite those first conditions in the tree shrew, that seems to be quite rare in terms of how the rhythm splits. Typically, we find that the rhythms split in response to high light intensity rather than a decrease in the intensity. And so this example here we see in a hamster, where we're seeing days exposed to light, and eventually, when exposed to light at a relatively high intensity, the rhythm will split. And note here again that the two oscillations have two different period lengths, but they soon match up and run parallel. So right when it splits, which is right around day 45 or so, you'll see that the one rhythm kind of parallels the same kind of delay, the greater than tau free run, but the other one kind of goes off to the left and shows a tau that's shorter than 24 hours, right? But quickly starts to revert back and slants to the right and parallels the other one, and they are about 180 degrees from each other, or 12 hours from each other. And this type of splitting has been seen in not just activity, locomotor activity, which we have here, but also in drinking rhythms and feeding rhythms, right? So we see an oscillation, a double oscillation in these behaviors as well. Now another example, departing from our typical mammalian models, this Texas spiny lizard had its pineal gland removed. The pineal gland is an endocrine gland found in the brain. That's important for circadian rhythms, keeping track of photoperiods, and by extension annual rhythms, and it produces melatonin, which is important for sleep and photoperiod. And so removing this gland from this individual showed a particularly dramatic effect of this decoupling of the oscillations. And specifically, they never ran parallel or recoupled at a 180-degree phase angle. So you can see that they're crisscrossed. One of the angles has a pretty pronounced delay, while the other one has a pretty pronounced advanced. So they're running one longer than 24 hours and the other shorter than 24 hours, completely independent of each other. So they've been decoupled and continue to run decoupled. And so these phenomena that I've showed you, right, the alpha compression, the splitting, and the after effects are not easily explained by a model that's built on just a single pacemaker mechanism, right? Especially the splitting here. And so that suggests that perhaps the apparent single rhythm is actually two separate rhythms, or at least two rhythms that are separable, that are most of the time coupled together, and that these two separate oscillations can free run with different periods simultaneously under specific parameters. And so these observations, especially, like I said, the phenomenon of splitting, led to the development of the dual oscillator model. And so the dual oscillator model states that circadian rhythms are generated by a system of two mutually coupled oscillators. And those oscillators have been called, or are termed M, the M oscillator, which stands for morning, and E oscillator, which stands for evening, because they're believed to be primarily influenced by light occurring in the morning or at dawn or late subjective night, right? Like 4 in the morning, late subjective night, and evening or dusk or early subjective night, right? Shortly after the sun has set. So let's take a look here and see if we can't break it down and sort of get a better conceptual idea of how the dual oscillation model applies to this, especially the phenomenon of splitting. And so it's proposed that each oscillator has a slightly different tau. The expressed single period usually occurs from a coupling of the two oscillators. So they're typically coupled, or they're like linked somehow. They're connected. That keeps them from separating apart. And the length, the tau, is a compromise between the two different natural taus of the individual oscillators. And that is determined by, or the single period length is determined by the individual period lengths and also the strength of each of the individual oscillators, the strength that they have on each other. So perhaps one oscillator might be more dominant, and it might have a greater influence over the overall period length when they are coupled together. So here I have just a rough image, a drawn image of a actogram of a nocturnal animal, and you can see here, for whatever reason, the single rhythm splits into its two separate oscillators. We have the evening oscillator to the left, because this is a nocturnal animal, and a M oscillator, the morning oscillator, to the right. And so we'll continue to look at this to understand what it's doing. So overt rhythms are actually regulated by both the M and the E oscillators together. So for example, in this nocturnal animal, the time of onset of activity is determined by the E oscillator, which I've highlighted in red here. So its position, its phase angle, determines when the animal wakes up and becomes active, whereas the time of the termination of that activity is not determined by the E oscillator, but it's determined by the M oscillator. So you can see they're kind of like, you can think of it as they're linked or kind of glued together when they're coupled, and they're kind of responsible for their respective portions of activity, in respect to into time, right? The E oscillator comes first, and then the M oscillator is responsible for sort of roughly the second half of the rhythm. So we already know from the previous studies, and then also our phase response curve exercise, that light in early subjective night, which corresponds to the E oscillator, causes phase delays, right? So it delays or pushes back those active periods to later in the night. And that light in subjective night, late in subjective night, or you could also say early subjective morning, right before the sun would be coming up, the light exposed during that time, which corresponds to the M oscillator, results in phase advances, moving the rhythm earlier. And so you can see here that both oscillators are kind of, in this model, independently influenced by the light at the end of the day and light at the beginning of the day in such a way where they're being pushed together. So, light bordering the active subjective night period is pushing these two oscillators together into a coupling phase in which they get linked, they stick, and then sync up and oscillate together. Right, so they're driven towards each other by the delays generated at dusk and the advances generated at dawn. And in nocturnal animals, the resultant movement of the two oscillators toward each other explains that compression of alpha during entrainment. So here we go. This is explaining not only the splitting, but also that compression that we've seen. You can imagine that the light, that dark period getting smaller, and the phase advance and the phase delay squishing into each other, causing a compression of alpha. So under skeletal photoperiods, which we've seen, or during exposure to an extreme T cycle, such as like a T equals 27 or T equals 22, they're driven so strongly together that the active phase could actually disappear. And this active phase is what falls between our two oscillators, which is what's being compressed together. So you can see here in this graphic, our two oscillators are being pushed together by light on each side until they couple. And then we have this skeletal photoperiod now that I have graphicked here. So there's just a burst of light at the end of the day, and then a burst of light at the beginning of the day, and the rest of it's dark. But that is enough to push these oscillators together, as you see here. And then, as we've seen as an example before, in real life, you see what's known as a phase jump, right? So eventually, these coupled oscillators are driven to a phase relation that is, intolerable, if you will, or it's just unsustainable. You can only push them together so much, like you can only push a spring together so much. And like that spring, one of the oscillators undergoes a phase jump relative to the other one to reestablish what they call a more tolerable phase relation between the two oscillators. These split phases will run at their slightly different tails briefly, but they tend to recouple. But when they recouple, they do so when they are 12 hours out of sync with each other, or 12 hours out of phase, or we could describe it also as 180 degrees away from each other, or a phase angle of 180 degrees, like we see in this hamster case here. And in fact, the M and the E oscillators are labeled here. So when they start to run parallel, they've recoupled, but interestingly, they've recoupled like completely opposite of each other. So initially, you can see here with the red lines, different taus. The E oscillator is running longer than 24 hours. The M oscillator is running shorter than 24 hours. but after a handful of days, a week about, they recouple completely out of phase with each other. Now, continuing the dual oscillator model, we can see that it also explains why light pulses during subjective dusk causes virtually instantaneous phase delays, while light pulses during subjective dawn result in phase advances that can take multiple cycles to complete. So the key thing to remember here is that the phase shift is measured by the onset of activity. At dusk, a light pulse causes an instant phase delay because the E oscillator is responsible for that onset of activity, and it's the one that is being delayed. So we measure delays by moving the onset of activity. What is responsible for the onset of activity? It's the E oscillator, which is where the light pulse is being emitted when the E oscillator is responsible for that activity. Alternatively, a dawn light pulse causes the M oscillator to advance, and in the process, because of that advancement, it actually undergoes a temporary decoupling from the E oscillator. Now, the E oscillator, which is unaffected by that light pulse because it happened too late, now has to advance in order to recouple to the now advanced M oscillator. And so this recoupling takes multiple cycles. It takes a little bit. There's a lag because it's not being directly affected by the light itself, but sort of getting pushed by the M oscillator. And depending on how extreme the phase angle is between the two oscillators will determine how much that lag is. So if you advance the M oscillator a whole bunch, that decoupling is the phase angle of that decoupling is greater. It's moved further out of sync, further decoupled from the E oscillator. So it takes a little bit of time for the E oscillator to advance itself to recouple to the M oscillator. Now, it's also thought that the phase relation or angle between the two oscillators is relatively plastic. So the two oscillators can couple together, but it's kind of flexible in terms of like how close and how far apart they are when they couple. And so this feature explains the observation of after effects, or transitions from different light phase regimes. So exposure to different phase lengths of light may decouple and then recouple the E and M oscillators at different phase angles to one another, which would result in different taus, free-running taus, as we see here, again, in this figure of a hamster. So for some reason, some mechanism innate to the two oscillators, when exposed to 18 hours of light, it decouples them and recouples them much closer together. And this might explain after effects that affect both the tau and also the alpha compression. And it may affect the tau by affecting which of the two dual oscillators is more dominant or influences the coupled phase more. This model might also explain animals that show distinct crepuscular activity or two distinct peaks of activity during their subjective night. It might be explained by that plasticity itself. And so animals that have this very distinct corpuscular activity might have a coupled oscillators that are relatively kind of far apart from each other. And so during early in subjective night, early in around dusk, it's largely the E oscillator that's driving that activity, and that fades out. And then before the M oscillator is further enough away that while it fades out, the M oscillator is not faded in yet, but eventually does by the end of subjective night or at dawn. The dual oscillator model also suggests the possibility that perhaps the E oscillator is capable of only phase delays and the M oscillator is capable of only phase advances, which would be a really nice way of explaining what we see in the phase response curves. That early subjective night is when we're sensitive to delays, and late subjective night is when we're sensitive to advances, which is where we find the E and the M oscillators respectively. However, multiple experiments in various animals using dark pulses during light, or bright light pulses during relatively dim light conditions indicate that actually each oscillator is capable of both delay and advances. Now, as a transition, because soon we'll be talking about our the suprachiasmatic nucleus. This paper, which was part of our reading by Wang and Barr, they discovered, in trying to understand the possible neural explanation for the dual oscillators, discovered some compelling evidence for sort of the material substrate from which these two oscillators might be generated. And so, as we'll go into detail, the neural basis of the circadian clock comes from the suprachiasmatic nucleus, or SCN for short. This is a very small piece of architecture in the very center of our brain, part of the hypothalamus. It's actually composed of two nuclei that are bilateral, so that means if you split our bodies in a mid-sagittal slice down the middle, you would have a left and a right suprachiasmatic nuclei on each side. But collectively, they're often referred to as the suprachiasmatic nucleus, even though it's two nuclei positioned right next to each other. And so it's part of a hypothalamus, right in the center of our brain, very, very small. And so in this interesting experiment, Wang and Barr, they had hamsters in which they were exposed to constant light, a light photo period, because they wanted to induce splitting. They wanted to decouple the E and M oscillators, which they did. And then they subjected hamsters to one of three possible manipulations. One was destroying, also known as ablating or lesioning the SCN. So they got rid of it. Alternatively, a control, a sham, they would go through all the motions of the surgery itself, everything except for destroying the SCN. So they would anesthetize, they would cut into the brain, but they just would stop short of actually destroying the SCN, right, to see if there were any effects of anesthesia or the surgery. And then a third possible manipulation, which was destroying only one of the SCN nuclei, only one of the suprachiasmatic nuclei. And so what did they find? Well, upon ablating the entire SCN, the hamsters became arrhythmic. Both of the uncoupled oscillations vanished. Right? So they performed these surgeries only after the oscillators were decoupled. So you can see in this middle graph, number B, there's a single oscillation in light. After it's about 2 months, the hamsters start to express 2 distinct oscillations of activity. At the red arrow, this is when they destroyed the SCN, and as you can see, it becomes relatively arrhythmic, the activity. Alternatively, when they ablated or went through the sham ablation, they didn't actually destroy the SCN. We see that the oscillations persist. Right? So controlling for those other factors, it does seem that it's the SCN itself that is responsible for the circadian activity. And the most interesting thing here is in hamsters where they destroyed only 1/2 of the SCN, only 1 nuclei, they see a correspondence where only 1 of the decoupled oscillations disappeared, whereas the other one persisted, which is what you can see here in figure A. So, with that, we conclude our overview of the dual oscillator model and our introduction to the suprachiasmatic nucleus. Next class, we'll talk about the search for the nucleus and its significance. So take a look at the readings, which have been posted on Google Classroom and also are posted on the syllabus. I hope you have a good weekend, and I'll see everybody next week.